I want to thank everybody present in person and also on the video cast for attending this afternoon's NIH Rector's Wednesday afternoon lecture series. My name is Charles Rotimi. I'm the scientific director for the Genomic Institute, NHGRI, and quite young in that position. I think definitely less than a year. So I'm still learning my ropes. I also want to acknowledge members of the NIH Genomics and Health Disparity Interest Group, co-chaired by Drs. Polly Joseph, Neil Hanchard, and Vince Barnum, who again nominated Dr. Wonkam for this year's awards lecture series. So now for the wonderful introduction of our esteemed speaker. Dr. Ambroise Wonkam is a professor of genetic medicine and the director of the McKusick-Nathans Institute and Department of Genetic Medicine at Johns Hopkins School of Medicine. After medical training from the Faculty of Medicine and Biomedical Sciences at the University of Yaoundé in Cameroon, he completed a thesis in medical science at the University of Geneva and a Ph.D. in human genetics at the University of Cape Town in South Africa, where he was before he accepted the position in Hopkins University. Dr. Wonkam practices medical genetics in both European and African contexts, and his research interests are reflected in more than 180 peer-reviewed publications, which are on molecular, clinical, educational, and ethical aspects of medical and human genetics. Very, very broad interest group. Over the past 5 years, Dr. Wonkam work has been supported by the National Institute of the NHGRI and also the Institute of Mental Health, and of course in the UK, Wellcome Trust, to study sickle cell disease and congenital hearing impairment. He also have active grants in coordinating consortia and also data center, and to investigate public understanding of big data. He has received many honors and awards, most recently the 2021 Allen Pfeiffer Award that honors a University of Cape Town researcher who outreach work has contributed to the advancement and welfare of South African disadvantaged people. Dr. Wonkam is Associate Editor for the American Journal of Human Genetics, the American Journal of Medical Genetics, the Journal of Community Genetics, and Academic Editor of PLOS ONE, and member of the Editorial Board of Human Genetics. He is also President of the African Society of Human Genetics and chair of the Steering Committee of H3 Africa Consortium, and board member of the International Federation of Human Genetics and the Steering Committee member of the Global Genetic Medicine Collaborative. And just on a personal note, I have known Ambroise since 2006, in the early days of our establishment of the African Society of Human Genetics, that I served as president for 10 years. And when I first met him, I knew he was going to be great in life. And he hasn't disappointed me or himself. And so, Ambroise, still very proud of you, and welcome.
Thank you.
All right.
Thank you, Charles. And I'm very grateful for your introduction. And you are probably one of the reasons I'm standing here today to speak to many of you, because I believe one of your brainchild, which is the H3 Africa Consortium, has greatly contributed to advance my personal career. For those of you in the room, I'm grateful that you are here today to listen to our talk. For those online, I acknowledge you, particularly those on far area of the world that have sent congratulations for this talk. I had the impression that I won an award. I hope to be the voice of all those people that seldom have a voice. Today, I will try to speak to my personal journey, career, and to convince you that African genomic variation is the next frontier of global genetic medicine. I wouldn't spend a lot of time on my training background. I consider myself as a global citizen, having lived and worked in 3 different continents under all the hemisphere. When someone asks me, where are you from? I say, I'm from Earth. When I try to set genetic medicine practice in Cameroon, my native country, I use genetic diagnostic before birth as a point of entry for genetic medicine. And this child here is the first child that was born in Cameroon after genetic diagnostic before birth for sickle cell disease. And when we set that clinic, immediately we started now expanding by practicing normal dysmorphology. The only specificity is that we will have more children with African ancestry with specific dysmorphic feature that will help the literature to better capture dysmorphology in most population in the world. Some of those specificity improve our understanding or our description of some condition. For example, I'm comparing here 3 kids with Williams syndrome from Africa who will look like any other normal kid in the street. But in a child with European parents, they will say, in what I have learned, that the child will have a full lip, what actually everybody has in Africa. We also extended our practice to single gene disorder, specifically for those that affect mental retardation and this large pedigree Fragile X syndrome in Cameroon. And one of our proud moments was to have established in that specific setting a multidisciplinary clinic for disorder of sex development. When we started the clinic, we quickly realized that we needed help for molecular diagnostic, for cytogenetics, but also for a surgical intervention was needed. And we called upon friends of us from Switzerland and from France, and to set the clinic. The clinic allowed over the first few years to have a clear description of disorder of sex development in that country, to have a molecular map, for example, for congenital adrenal hyperplasia in all the region of the country, to provide a treatment for some children that without genetics and a combination of multiple specialty across numerous countries that they wouldn't have had otherwise before. And also for some condition, very complex condition, like in this boy, 14 years old at the time we saw him, he have a true disorder of sex development, one ovary on one side, a testis on the other side. He was self-identified as a boy, socially, legally, and also psychologically, but he had a female chromosomal composition at the time. So after all the appropriate counseling, we were able to provide him with a mastectomy. He had a very strong breast development at the time, remove the ovary on one side and provide the necessary hormone for his growth. This experience allowed us to send an opinion piece at the time when the International Federation was using genetic for gender testing to advocate against the use of it. And probably this is perhaps one of the most influential piece of work we ever published. According to the American Association of Genetic Counselors, they thought that this piece of work advised the International Federation against the use of gender testing for sports. That specific program and a few examples that I show also support the fact that rare disease, those conditions that are hidden in family, is not just a consideration for a wealthy nation. You can see it everywhere. Here is one of my cousins in another time. His name is Snape. Snape lived about 4,000 years ago, and Snape had achondroplasia. Snape was a priest at the court of a pharaoh. Snape was also the responsible for the wardrobe at the pharaoh at the time. Snape had a very beautiful wife, as you can see here, and the wife had a normal size, and they had 2 children. This single slide shows that maybe 4,000 years ago, 5,000 years ago, first of all, rare disease was still there. And secondly, our tolerance for difference was probably much better than today. We moved from Cameroon to South Africa about 13 years ago. And in South Africa, we landed in a country where there was already a relatively established medical genetic practice. It was a country also with extremely pathologic history. At the center of our university now, we have what was initially called the Jameson Hall. We changed the name of that hall to the name of the lady that is here on the screen. That lady was from the Khoisan ancestry. She must move from South Africa some 100 years ago to Europe, exposed in museum to be seen and be paid off like an animal. He eventually died in France. His remain under the UNESCO was bring back to South Africa recently. This introduction is not random. This introduction was to show that in that country with such pathologic history where numerous group was separated, it also shows a very different profile of genetic condition. I have worked at the University of Cape Town where Professor Peter Byton established the genetic clinic. The specificity of practice in Southern Africa, and South Africa specifically, is the prevalence of founder effect because of the separation of population, and also the way the population was established in South Africa. So we will investigate genetic condition according to the ethnic background of people. For example, in Europeans with Afrikaner ancestry, hypercholesteremia is the most common condition. And you also see the difference of allele frequency of mutation according to the ethnic background in this picture. Because population were separated very well, there was also there's a high level of founder mutation, which is the hallmark of single gene disorder of South Africa. We described the founder mutation for Bardet-Biedl syndrome. We recently described one for Usher syndrome, specifically in population in South Africa with black ancestry. During the apartheid system in South Africa, in a wine farm, worker was being paid at the time with wine, actually the leftover of wines, make fecal alcohol syndrome the highest condition with environmental etiology in our practice, 7 out of 1,000, the highest in the world. So again, I'm sharing this example to show that if you move across the continent, according to the country where you are, your practice will be relatively different. We investigated sickle cell disease in South African black population. Actually, there is no sickle cell disease in South African black population. We were a bit surprised. We tried to look at the haplotype and other malaria-resistant genes in Southern African population, including among Zimbabwean. There is clearly an increasing gradient when you move from Southern Africa to Central Africa. We also realized that actually South African black population had sickle cell disease in the past because we could see some indices of the haplotype around the sickle cell mutation. But having moved in South Africa more than 1,000 years ago, and where there is no malaria, there is no more need to keep the mutation. And this is not a South African. This is very much my son. He was 20 years younger in our Geneva apartment. What you see in the bottom left is the umbilical cord that link my son to his mom when he was born. And we kept that umbilical cord and buried it in one specific place in Western Cameroon where my parent and my four parents were born in a ritual way. And when I visited the house of Nelson Mandela and Winnie Mandela in Soweto, Vilakazi Street, behind the house there is a tree. Below the tree, there is a voice on Winnie Mandela saying that under that tree, they bury the placenta of their children. The exact same practice that we do up there. What does that mean? It means that Bantu from the middle of Africa and those in South Africa have nearly the same way of speaking, the same culture, but they do not have the same genetics. And this is critical when we are investigating population in Africa. In middle of Africa, you have sickle cell. In Southern Africa, you don't. In Italy, you have sickle cell. In Southern Africa, in black South African, you don't. That means that technically, a Bantu guy from Yaoundé is closer to an Italian guy genetically for sickle cell than to the South African blacks. So there is a clear dissociation between anthropology, culture, and genetics. And the way we call people cannot be Bantu alone. It needs to be Bantu from a specific region if we need to apply that genetics to medical practice. Our understanding and better understanding of this morphology of numerous population allow us to be linked to Max Mwanke. I can see Debo in the room. Debo actually did the connection. And we introduced a specific issue and a specific section in the American Journal of Medical Genetics called this morphology in diverse population, and that section still exists. And the goal is clearly to give the opportunity to all those phenotypes that are not necessarily seen, that may be specific, to have a space to easily publish it. I'll now move to a clear research agenda that was informed by our experience working in Africa as a medical geneticist. And to say to people here that are funders, actually there is a 3 reasons why we should massively invest in African genomic variation. The first is ancestry, we are all African. The second is ecology, and the third one is equity. On the right side of this slide, the first person is Lee Berger, the second is the president of South Africa, Cyril Ramaphosa. They are both doing one thing. I think they are in a competition of kiss with Omona Lady. And Omona Lady is one of those that was discovered in South Africa. The reason I'm showing this is that not only we are all African, but there are some humanoid that never move out of Africa. And that there are some real evidence that we all have up to 2, 3, perhaps 5% of DNA content of those humanoid that we call ghost DNA, for which we are not yet able to read very well. I'm saying that investing in African population will allow us maybe to explore those better. Why do we need to explore it? Let's come back to COVID-19. COVID-19 have not been that bad in Africa for numerous reasons. One is the youngest population. The second reason is this marker that was described by Dr. Pablo in population of European and Asian ancestry that was inherited from the Neanderthal that increased higher susceptibility to COVID-19 with people of European parent. So investigating those old DNA will be important for health, it will be important for disease. It can be best investigated in population with African ancestry. We also know that the current reference genome do not capture variation, all variation in population ancestry. In this study, 10% of DNA variant was not captured in only less than 1,000 African with Caribbean ancestry. The work published by the H3 Africa Consortium also show at least 3 million single nucleotide variants that were not referenced, and an evaluation shows that we still have some room to discover some of those new variants. By not investigating the population of African ancestry, we are shooting on ourselves because on the 2.5% of genome-wide association data that has data from population with African ancestry, they account for 7% association. And the reason of that is that the shorter haplotype amongst African make fine mapping a little bit more easier. And even if you use whole exome sequencing to investigate complex trait, it was shown by investigating schizophrenia that there were rare damaging mutations in about 1,000 Africans from South Africa. Those rare damaging mutations was replicated in a Swedish cohort of 5,000, but African group yielded much more largest effect size because of high level of variation. So it all works, both for single gene disorder, but both for complex trait. And we all know this cartoon boy of this type of analysis comparison, which is one variant that is 200 times more common in population with African ancestry than in population with European ancestry, and responsible for 40% reduction of cholesterol, and that have led to new medication that we all use. This work from Charles Rochimi-Hayes' group also discovered one gene that is monomorphic amongst European and Asian, but responsible for type 2 diabetes mellitus. What does that mean? It means that you will have investigated 2 million, 3 million, 10 million of European or Asian, you wouldn't have found it unless you investigate a population with African ancestry. I will now move to a single gene disorder. We know that the profile of single gene disorder is also different. For example, Huntington disease. If you have Huntington disease with someone with European ancestry and test for HTT, that will explain all the cases. But it will explain only 70% of cases with Huntington disease with African ancestry, and 30% will be explained by a second gene. Our own work on hearing impairment have shown that GDB2 that explain 50% of congenital hearing loss in most European and Asian explains 0% in most African. There's a few exceptions. I will speak to it a little bit later. The second reason we should invest in African genomic variation is ecology of Africa. The north-south axis of the African continent make that you have variable level of climate from the Mediterranean area to South Africa, different type of food, but also different type of infection along the way. And these infection, food, and environment are mode of natural selection. We all know the story of sickle cell disease. We know the story of APOL1 selected against a trypanosome. We know other story of some variation that was selected in Africa for which we do not yet have the reason. For example, in the H3 Africa Consortium, there was 65 more loci subject to natural selection that still need to be explained. Of course, those variations sometimes have relation to disease like APOL1 and kidney dysfunction in population with African ancestry. I hope because of those reasons, ancestry, we are all African, and also ecology, studying African population is an imperative. I will now speak to the imperative of equity. Africa represents 15% of the world population, but technically 5% of the world domestic, gross domestic product. That means that in Africa, even if we have all the variation possible in our DNA, we may not always be able to investigate them because of the poor investment in research. And as a consequence, an African investigator will only perform his research properly if he's linked to an American or a European institution, even for infectious disease as shown on this slide. As a consequence, our research priority may not be the research priorities of funders. For example, in this slide that we published 10 years ago, most population genetic performed in Africa are population genetic study, but not necessarily a study on conditions that matter for us as clinicians like hemoglobinopathy. And the knowledge amongst physicians also is not really optimal because they are not exposed to genetics in practice. The knowledge in the general population is not optimal. For example, in this study in Western Cameroon, we show that in this small family with Fragile X syndrome, it was called a curse because in a royal family where there was a huge amount of Fragile X syndrome, it was believed that the founder of that village, a Chief K, threw a curse to his wife because they did not mourn someone with intellectual disability from that village. And then he said, all your son will have intellectual disability. But actually, it was not the case. It was Fragile X syndrome. The collaboration with Africa have a strange way up to recently with not non-inclusion of African center in publication, non-inclusion of African authors in publication. But things have changed over the past 10 years with H3 Africa. And these big programs have allowed not only to have a better knowledge of the structure of African population using DNA and genome for population within Africa, it have allowed African investigators to be very present with high-power publication in the literature. He has allowed African PI to train generations or the next generation of scientists. I'm very proud personally to see in the room Samuel, Edmond, and also Cedric that all trained in my research. And if they are here at the NIH, that means that we did a good job. And these are a few examples of people that have their PhD in my group, and many of which are doing their independent career across the world. This would not have happened before the recent investment. I will now use two examples to show how we can harness African genomic variation, but at the same time address the issue that matter for population of African ancestry. I will start with the tragedy of a common sickle cell disease. Described in this country about 112 years ago, we know that most people that evolve in sickle cell disease, at least from my perspective also, introduced me to genetics. I'm sharing with you a private email that was sent to me by Hek Kazezian in September last year before he passed away, where he was reminding me that by joining Hopkins, I was joining a specific lineage, and that I was actually his great-grandson scientifically, and he also had the other people in the lineage. And so I went back and redrew the pedigree and add every single person he requested. Unfortunately, when I joined Hopkins in January, he was gone. And funnily, I'm sitting in his office at the moment, so the history will continue. Sickle cell disease will evolve in Africa as per Charles Rochimi, his work 7,000 years ago. And this is a condition that distorts red blood cell under hypoxia. And those distorted red blood cells obstruct the vessel, and as a consequence, you have organ damage, anemia, and also susceptibility to infection. Since 112 years ago, there is not yet really an available definitive cure that is really equitable. We know there is bone marrow transplantation and most recently gene editing and gene therapy. But in Africa, half of children with sickle cell disease will still die before year 5. In America, the situation hasn't changed dramatically. Adults with sickle cell disease die in their 50s. That means that in all parts of the world, we still need to look for a solution for cure sickle cell. In America, for example, people with sickle cell disease will die before of cardiovascular complication, as shown in this slide. And all these cardiovascular complications are subject to genetic variation. So I propose that we can use sickle cell disease at 3 levels, genetic at 3 levels for sickle cell: primary prevention, secondary prevention, and therapy. Primary prevention is only genetic before birth, and this poses a lot of ethical issues. As per our own study, in at least 3 different African countries, we show a potential conflict amongst different layers of the population. The doctors are not comfortable at all with genetic diagnostic before birth for sickle cell disease, which is not the case for the parents with affected children, as well as our adult patients. So we thought that moving to use genetic for secondary prevention would be a good approach in research. The reason why we have invested this day to look for susceptibility for complication with sickle cell disease. This is the case for kidney dysfunction, where we show that 3 genes, APOL1, of course, alpha thalassemia, and HMOC1, explain most susceptibility of kidney dysfunction in sickle cell disease. And for therapy, since about 40 years, because of the cooperative study that was performed in this country, high level of fetal hemoglobin was associated with longer survival. Fetal hemoglobin is a quantitative threat subjected to genetic variation, and this knowledge allows Dr. Stuart Hawkins, his group, to show that by removing the transcriptional repressor of fetal hemoglobin, that is BCL11A, you can cure a mice for sickle cell disease. Everybody was very excited because of this news, because we thought that was the best target for sickle cell. Until someone realized that there are real patients in real life that have a deletion of BCL11. These patients have increased level of fetal hemoglobin, but they all have some level of intellectual disability and brain malformation that make BCL11 at that time nearly undruggable. Again, Dr. Hawk in his group shows that there is a transcriptional, an enhancer of BCL11 that is specific of red blood cell. Otherwise, it modifies the expression of BCL11 only in red blood cell. And by changing variant of that enhancer, you have the same effect as if you have removed BCL11. And in summary, you remove BCL11, you have a child cured with sickle cell, but unhappy because of neurodevelopmental condition. You modify a little variant, 50 kb before BCL11, you have increased level of fetal hemoglobin without those malformation. And based on that, they are successful gene editing for BCL11 for sickle cell disease, one that was published only this year. But our study in Africa and those of my colleague Julie Makani in Tanzania have shown that those 3 variants that explain up to 50% variation of fetal hemoglobin in people with sickle cell disease in Europe, in Europeans rather, explain maximum 20% of variation in African. So I thought that maybe we did not find the other missing irritability because the GWAS array used to perform this study was designed on European background. Using the advantage of the array developed by H3 Africa, we rerun the GWAS for fetal hemoglobin in a group of sickle cell disease from Cameroon. We found at least 3 new loci that explain fetal hemoglobin. One very strong candidate is here on chromosome 15. We know the genes that is explained. We are now doing some cell experiment to show the mechanism of that. We have done a meta-analysis between African cohort, the published data from Julie from Tanzania, and also graciously all investigators in this country have given us access to data from African American. And our meta-analysis of all those data show very strong, not only connection with the variant we found on chromosome 13, but 3 new variants that will be explored at target of therapeutic. I hope to request your pardon not to show too much detail on this, and you'd understand why. We need to do that because this is Grajevis. Grajevis is a real patient living with sickle cell disease. He's one of our patients from South Africa. And in this nature he's telling his own story, his life story, and his hope as aspiration that researcher will be able to make gene editing, gene therapy affordable, equitable, specifically for those people in farthest part of the world in Africa that really need it. One thing that have not yet been properly investigated is other variants that may modify the expression of sickle cell disease. And in this initial study that we perform, we look for those rare mutations that may be associated with long-term survival of sickle cell disease. Maybe one important findings here was that in the long survival, we found genes that lower blood pressure in sickle cell disease, but also some genes that increase the level of L-glutamine. In other words, it means that some of these variants potentially increased endogenously the level of L-glutamine in those patients. We will extend this study a little bit in more patients. We have more than 2,000 patients and data to reconfirm this data. Why is it important to look for alternative route for modifier? Again, this is another cousin of mine. I have Nubian ancestry, the reason why all Egyptians are my cousin. So this guy is Tutankhamun. Tutankhamun actually had sickle cell disease because of many studies that was recently performed, microsatellite study, but as well as the MRI of his bone here that show osteonecrosis that is specific to sickle cell disease. Tutankhamun lived about 6,000 years ago. That means that for at least 6,000 years, sickle cell disease might have put some specific pressure on the genome of those that have the condition, and perhaps we have some marker of selection due to sickle cell itself. We tested that hypothesis in a very small cohort and show at least 3 regions that are found to be selected in patients with sickle cell disease, but not in control in the same population. And we will extend this data because at the time we performed this, we did not have the necessary power, and we should be able to show more on this. Why do we need to look for those markers of selection? It is possible that many of those markers co-evolve at the same time. This is a simple study that one of my young graduate students performed. We look for data point of 3, 4, at least 4 variants that have been selected in Africa. One is the sickle cell disease, the other is alpha thalassemia, the third one is G6PD, and the fourth is APOL1. As you can see, most of those data points plot exactly the same region. It suggests that someone like me who may have all those 5 variants in his blood, individually each of the variants has some effect on health and disease. How if we combine all of them? I'm inviting a young mind, one young African mind, mathematician, to design new ways to interpret this. For example, for genetic medicine, the interaction of this variant and their impact on health and disease of people with African ancestry. We believe to have the tools to do it. We have set a cohort of nearly 15,000 patients of sickle cell disease across 7 different African countries funded by the NHLBI. We are in the second phase on that study, and hopefully in the next 5 years, we'll be in a position to address some of those questions across the continent. My second story to explain why investigating African genomic variation will inform health for people in Africa, but globally, is the story of hearing impairment. And I call this second story the case of a silent epidemic. Hearing impairment affect in America at birth about 2 in 1,000 child. In Nigeria, about 7 in 1,000 child. In South Africa, 6 in 1,000 children. So, hearing impairment when congenital, when a child is born deaf, 50% of the time, this will be due to a genetic cause, and most of the time is non-syndromic. Our data shows that for those children that have hearing impairment with something else, Waardenburg syndrome, which associates potentially hearing impairment and some pigmentation defect, for example, striking blue eyes in this broad ancestor, and the expression is actually even variable. is the most common syndromic hearing impairment in population with African ancestry. Our study were both from Cameroon, and South Africa, Mali, and Ghana. Data from our group and people else shows that GGB2, we spoke to it before, do not explain hearing impairment in most population of African ancestry. We found one exception in Ghana where a founder mutation will explain one family out of 3. Because that was a missense mutation, Samuel in the room developed a rapid test for that variant that can be implemented very easily. It can be tested in any kitchen in Ghana. We worked with the Ministry of Health in Ghana to integrate that rapid test to the newborn screening program in Ghana, a real proof of easy genetic medicine implementation. We also took the opportunity to explore why that mutation and when that mutation occurred in Ghana. And we found that the mutation occurred in Ghana about 11,000 years ago in a single Ghanaian ancestry. The same mutation was described in Japan. Our investigation shows that our mutation in Ghana rests on a completely different haplotype than the one found in Japan. And the one in Japan was explained by an age of 6,000. So it's much more younger. And of course, I think I spoke to this already. It was found to be age 11,000 years. And why would someone select this variant over time? A study in Ghana shows that when you have that variant, you have a thicker skin. And is this thicker skin protect you against the mosquito bite of malaria? We don't yet know. And this data was also confirmed in population with American ancestry with that variant. These are very recent data that we performed in Senegal. In Senegal, we found a second exception, a missense mutation that explained 25% of hearing impairment, congenital hearing impairment in GG2B2 in Ghana, in Senegal. But surprisingly, we also found a Ghanaian mutation. I've shown the map of Africa because to show that Ghana and Senegal do not share border. We have data from Mali. There is no GGB2 mutation in hearing impairment in Mali, and Mali share border with Senegal. We have data from Nigeria. There is no GGB2. How is that we have that mutation, Ghanaian mutation, that jump all those countries and end up in Ghana, in Senegal? We think that this happened through slave trade, because slave was assembled in Gore, and then from Gore shipped to America. And actually those two mutations, the Ghanaian mutation and what we call now the Senegalese mutation, are found amongst African American. And moment, one of our fellow is trying to prove that at the molecular level. And the way we're going to prove it is to show that the variant, the Senegalese variant found, the Ghanaian variant found in Senegal rests on the same haplotype. We use exact same study to investigate the age of the Senegalese variant. If we exclude those GGB2, very few African countries have investigated hearing impairment at the molecular level using, for example, whole exome sequencing. Only 13 out of 54 African countries. That means that we still have a lot to discover only for this condition in Africa. Why do we still have a lot to discover? Because if you use targeted gene sequencing, that use 100 genes of hearing impairment, the pick-up rate amongst people with African ancestry is very low. That means that we are not looking at the right genes. There are many other genes that still need to be found, including among African American, where the pick-up rate will be 26% compared to 70% of American with Middle Eastern ancestry. And if we look at the transcript of the inner ear, many people think that we still have at least 1,000 hearing impairment genes to discover. And I will argue that the right place to discover them is in Africa. The reason that most rediscovery of recent hearing impairment was in the Middle East and specifically in Pakistan, where 40% of hearing impairment genes were discovered because of the consanguinity. In Africa, because of the diversity shown here by numerous studies, we sample a lot of family with at least 2 children that are born with hearing impairment and investigated using whole exome sequencing the origin of those hearing impairment. Of course, we started by those rare genes, those ones that were explained, found only in one family, for example, CLIC5, and found a second family in Africa, which means that we contributed to global knowledge by confirming those genes. This is another example on one family that initially was described, the first family was described in America only 15 years ago, and the second family found in South Africa rest one of the hearing impairment genes. And we also take the opportunity to investigate new genes, and these are data from Ghana. And in Ghana, we found 7 candidate novel genes for hearing impairment. Some of those candidate genes was, I learned something recent here, was obvious. For example, in this family, small family, with Waardenburg syndrome, there was a PAX8 mutation that Mendelized on the vertical manner, and there is a PAX negative mice, and the PAX8 negative mice is deaf. And functional analysis from others performed already nearly 20 years ago has shown that. Another example, this is a family, small family in Ghana, where we found mutation in SOX9, and there is a mice model negative for SOX9, KO SOX9s, who have malformation of the inner ear. The other genes, we are currently developing 4 mice models to show the implication of hearing impairment of the other genes that we describe in Ghana. I can tell you we have the same number in Cameroon at the moment that we are investigating. Something that we were not expecting, we took a random approach of exploring genes that we know are affected with hearing impairment in the same country, patient versus control. In principle, if you assume that this is a single gene disorder, you shouldn't see a major difference in minor allele frequency in those genes, patient versus control, but it was not the case in this study. We found that a derived versus ancestral allele in this gene with hearing impairment was different, a patient versus control, suggesting at least 3 things. The first is that even if you have a single gene, maybe variant in the other hearing impairment genes are modifiers. The second possibility is that maybe there is a good reason why some of those genes were selected over the evolution. We don't yet know why. And the third possibility is that by investigating hearing impairment genes, we may perhaps understand why humans speak, because actually if you don't speak, you will not hear. I'm here inviting us to invest in molecular evolution of hearing. This was just a very, very small study. Actually, the journal liked it so much that they make the cover for it. So this small example showed 2 things that can be applied to many other single gene disorders with genetic heterogeneity. First of all, very few people in Africa have been investigated for this condition called hearing impairment. The second, there is a high, using whole exome sequencing, we can explain most families at the level of 90% resolution rate. For those families that are not resolved by whole exome sequencing, our discovery rate for novel genes is 15%. Most of the variants in non-gene that we found are novel variants. 80% of them have not been associated with hearing impairment, even in non-genes. That means that we will, by using those variants, improve the disease gene curation globally. Of course, by generating this data, we will face a few ethical issues, and one of them will be the actionability of some of the genes on which one to explore. Some of them will be, should we even revert or return the result to those families that we have investigated? Other is that even if we return those results, would there be any practical implication in terms of care for those families? Are we not creating additional problems? This is some of the questions we are investigating in one of our projects called IFGenera, that explore how we should return results, what should we return in Africa, and what is the process for returning results if we use next generation sequencing to investigate families. This is one of the meetings of a group that we convened in Tunisia to explore some of those questions in 2019. We hope that the data we are generating, for example, for genome of exome, will allow a better representation on the database in the literature, and this will improve our disease curation for anyone else. This is another example why we should improve the database. That was a variant for cardiomyopathy that was labeled pathogenic just because there was not enough African in the database, and that variant actually was present in 30% of people with African ancestry. And a simulation shows that even if we included only 50 people in the database with African ancestry, this mistake would not have happened. It means that this beautiful home proposed by Dr. Collins with genomic medicine resting on the Human Genome Project still has a few cracks in the foundation. The missing variant in the first genome of the oldest population with African ancestry. I have proposed last year that if we need to address this question, we need to sequence at least 3 million Africans from various parts of the continent. Initially, this proposition or project of idea was just a crazy idea that hopefully have found a lot of attention across numerous offices, and maybe only next year a version of this project will see light. We believe that if we want to make genetic medicine truly equitable, we need to have a representation of our genome, and we need to start with our common ancestral genome of population of African ancestry. Only we do that at scale, only we do that, we will make genetic medicine truly equitable, improve scientific knowledge for all of us, but also address equity of and access for all population. This is a game I would like to play. This is my plea for the youngest of us in the room. This is my hope and aspiration for the next 10 years of my career, the 20, 30, maybe 40 years is I'm still around. And this is my invitation for you to join us in this journey of exploring our common genome for population of African ancestry. This is the reason many funders I would like to acknowledge here have helped supporting some of the work that I'm presenting today, and I'm thanking you for your kind attention.
The H3 Africa has brought much, but in contrast to the accessibility of NIH and British Biobank data, access to H3 Africa seems to be the subject to the gatekeeping of the project's PIs who first want to mine the data for their publications. Do you have any comment on that? There are several companies that are available for bioinformatics and follow-up genetic studies, and including my own, this person, and wondering how you see the future.
Yeah, I think first of all, I would like to thank the colleague that asked the question for his positive comment. The second, regarding H3 Africa data, the H3 Africa data definitively at the IGA, they are accessible. So the comment that there is a gatekeeping is not fully true. Mm-hmm. The second is that there have been, during the H3 Africa development process, a window that was given to investigators to first explore their data. That window was exceptionally long, 24 months. But I would like people to put this into context. Mm-hmm. If you are working in Africa, and I've worked in Africa for the past 15 years, if you have exome data that you have to sequence out of Africa, just the transfer of those data from the core facility where you sequence to Africa take many days. And sometimes you have electricity that are cut and that delay your data. If you want to analyze data, you need to have an army of bioinformaticists that allow you to do so. You don't necessarily have that army of bioinformaticists. That means that you are competitively disadvantaged against anyone else, for example, an investigator in this room that have army of postdocs that will analyze. So one of the idea of giving 24 months leverage was to allow investigators to have a chance to explore those data, taking in account the behavior of international scientists that have performed helicopter research for the past 20 years in Africa, taking in account the lack of infrastructure and human power that may have allowed investigators actually to have access and ownership on the knowledge they have generated. And anyone that cannot take that equity principle in account will be misguiding. But I must say those data definitively are available, and we are much, much encouraging that all the data that we generate are in the public database.
Well, that's a great perspective from someone who clearly knows it so well. Any questions from the?
Yes. Thank you, and good afternoon. Thank you for such a wonderful lecture and educating us on this topic. My question is two parts, and you somewhat ended on a component of my question, but I wanted to ask Early in the lecture, you referenced the existence of 3.4 million SNVs, and I wanted to ask you that in the yet required description, but could be harnessed to elucidate unknown areas of the genome. So how do researchers approach a task that appears so vast? And then from there, how is equity in the questions asked and the outcomes of that research ensured?
Please, can you repeat the last part of the question? Yes.
Yeah, how should researchers approach a task that appears so vast? And how can equity in the research pursued and the benefits of that research be ensured?
There are many layers to the question. Maybe I'll start to speak to a conference that was given by a very famous writer from Nigeria. Her name is Chimamanda Adichie. She has a very famous TED conference that she called The Danger of a Single Story. And in summary, what Chimamanda says is that the single story is not that the story is not untrue, it's just that the story is not complete. What we are trying to say is that what we have in the database there is not that they are untrue, no. Everything is correct and everything can be used for any population mostly, but they are not complete. That means that half of the story cannot be told. So that means that for some population that we have, probably we need to address the question of equity, increase the representativity on variation. For the question of equity, we should extend not only to the representativity of variation, but also to the representative of those that investigate those variations. I do think the workforce needs also to be from all parts of the world, because there are some questions that you can only address because you have a specific living experience. I met with a couple of students, post-bac students before, and one of the advice they asked, and I tried to tell them, is that you need to get connected to the society, because some of the hypotheses of your research will be from the living experience. And I gave one specific example. We start using whole genome sequencing to investigate sickle cell disease because one of our patients, who happened to be one of our friends, say, why is that I live 65 years old, having sickle cell disease, living in Africa and without treatment? And I thought that perhaps this guy may have some of the variant that if we do not have that direct contact, it may not have help. And that is living experience. So increasing the pool of scientists, data from every part is not just representation, it's also increasing different mindset, different way of seeing life, and different way of seeing science.
Ambrose, sorry, I have a question, maybe a couple of questions actually. Regarding return of results, I was wondering how we can guide against expectations of families when we return these results to them. For the first time, perhaps that's when they will know the diagnosis of what their children are suffering from for all these years. How do we manage that expectation of, would this lead to a cure? Would this lead to treatment? I'm concerned about that. I don't know if you can make one or two statements about how we can monitor and manage that expectation. So the other question I have is, I was actually quite intrigued when you made a statement about sickle cell was in South Africa at some point.
Yes.
But he left.
Yes.
And I wonder how that data was determined, and because it's absolutely fascinating that he was lost.
Okay. I think the first question, there are two ways to respond to it. The first one is real data. When we perform our If Genera project, we use 3 different case scenarios. We investigated using qualitative approach, would you like the result to be returned in 3 groups of people? The first one was people with sickle cell disease. The second one was people with Fragile X syndrome. The third group was group of people with neurodevelopmental condition. The fourth group was people with infectious disease, specifically HIV. In our group of HIV that was investigated in Botswana, we interrogated parents whose children was affected by HIV and in which there was some genomic research. They want the result back. Even though they will not cure their child, their reason is that they get educated of what had been researched. In the group of people in South Africa that were investigated, all these data I'm talking about are published. So if you could type on the internet, you're going to see all those data. In the group of people with neurodevelopmental condition in South Africa, again, their expectation of having the result was not to cure neurodevelopmental condition in their kid. Their expectation was a teachable time with those people. Their expectation was also a diagnostic closure. We saw the same in Cameroon with Fragile X syndrome, where people were just happy to know, to have a name of the condition that their child have. And so we think people want the result back. That's the first thing, whatever they will use the result for. The second part of your question is that can we act on the result that we have? During the meeting we had in Tunisia, we arrived to the point that action on the result may not be necessarily medical care, it may be counseling. And that group of experts resolved, for example, that people that have heterozygosity of S, carrier of sickle cell disease, they should know, because that can inform their reproductive future. That can inform the choice of partner, and that is return of result and actionability. There was a question, for example, for breast cancer genes. If you are in a place where mastectomy cannot be performed, should we return the result? The response of experts was that the return of result should not be dictated by the availability of medical services. The patient, first of all, want to know is the first step. The second step, the patient can have the possibility to move to one specific center where action can be happen even in the same country and sometime internationally. So this idea that people would not like to return of result, our evidence from study on the ground says that they want the result to return. How do we action on that is a different question. The question of the sickle cell disease, how does it get rid of in Southern Africa? Many geneticists say sickle cell is a so-called balanced selection. If you have sickle cell, you die of sickle cell if you are not treated. If you do not have sickle cell trait, you die of malaria. So at the end of the day, people who have heterozygosity of S survive. What would perhaps have happened in South Africa is that because they stayed in an environment where there was no malaria, progressively those people that have sickle cell disappear, and those people that have AA do not have survived because there was no malaria. And progressively the threat was not in the population. How do we convincingly show how it happened, when it happened? That would be a question that you and I should sit and brainstorm on. And I think that brainstorming will allow probably having sample across the gradient on frequency of sickle cell. So from South African Bantu, Bantu Zimbabwean that have LB less, Bantu Zambian, so upwards. And actually by studying variation in all those around the sickle cell mutation, we may know perhaps at what time we were less and less having the frequency in the population.
There's one more important one here, but you go first.
Okay, well, I think we're about out of time. I can just ask after the session if that's helpful.
Let me turn this on. Okay, well, we had lots of questions online, including one from someone with a similar last name as yours, and I think he can ask you later. I think it was your son. But here's a good one. How much East, West, South, and North African genome is represented in the current genome-wide studies?
Excellent question.
Yeah.
Very, very good question. The straight answer is that I don't know. And the following answer is that we have to do it. I think maybe I'll go back to why we need to do it and sample. Let's say African population is not enough. We need to sample according to region. We need to sample according to country, and even within the same country, different population. One implication of Dr. Ruchimi's study on the age of sickle cell mutation that have not yet been explored. Sickle cell, if sickle cell have a single origin in Africa, and it does have a single origin in Africa, there are these two studies on that. That means that all sickle cell patients in the world, they all came from Africa. We have sickle cell in Italian, sickle cell in Greece. Actually, the first population screening of sickle cell in the world started in Greece. We have sickle cell among Portuguese people. We have sickle cell in the Middle East, in Indian people. One question that have not been explored, which seems to be very strange, is that when does sickle cell arrive, for example, in Sardinia, in Italy? At what time? because they all come from Africa. And if it is sickle cell date 7,000 years ago, that means that someone have walked or run or take a car from West Africa to Greece at some point of time. What question we should then ask is that at this time in Greece or in Italy, Sardinia, there is no more malaria, right? Sickle cell probably have persisted because of reproduction and survival. In a different environment where there is no more malaria in those places, how does expression happens and so on. So there are numerous layers. Again, let me come back to the sickle cell again. If that person walked to Italy, he probably crossed the Sahara Desert. He probably crossed the Mediterranean, and he swam, right? Moving maybe 4,000 years ago. That means that he left behind, for all those populations in those areas, some trace of his variations. And those areas have different environments. So we have to capture both the variation and the effect of the environment. We need to sample everywhere in Africa, including Northern Africa. There was a private conversation earlier before this talk, is that maybe one of the things we should have done if H3 Africa was to engage more North Africa and to have more variation in those areas and to understand what is the connection within the continent. There is a Egyptian genome project that shows that actually present-day Egyptian, whatever how they look, have 40% about their DNA variation that come from West Africa. But if you go to Egypt today in the Delta area, you will have most impression that most Egyptian maybe will have Arab or ancient Italian ancestry, which is not always the case. So there's still some missing link of the mapping of variation within the continent that we have to do, hopefully in the next 10 years, perhaps 20.
Well, thank you so much, Dr. Wonkam, for the wonderful presentation, and everyone that has been able to come in person as well as online, and we will get to some of your questions. So thank you very much again, and we will just stay a little bit afterwards to take some pictures and to present you with a plaque. Thank you.